Hello, this is Dr. Sage again. Today what I want to do is I want to go through several practice genetics problems to talk about more different types of problems that you might see on a genetics exam. So the first thing we need to discuss is how you solve genetic problems in general. For example, during the lectures I taught you how to solve a monohybrid cross. That's where you have one gene and you would do that standard 4-square Punnett square to solve it. I also taught you how to solve a dihybrid cross. That's where you have two genes. That's where you're going to FOIL both parents and fill out that large 16-square Punnett square to solve it. Okay. Well, that's if you have one gene or two genes. Okay. Well, what if you have three genes or four genes? Hypothetically, you could figure out how to FOIL when there's three genes or four genes, and then you could fill out a Punnett square. But if there's three genes, I think that would be like a 64-square Punnett square. Four genes, I believe that would be a 256-square Punnett square. You are obviously not going to fill in a 256 Punnett square. So how do you solve it if there's more than two genes? For example, let's say I tell you that you have these two parents. Okay, and one parent has these four different genes, so the A gene, the B gene, the C gene, and the D gene. And that one parent is a heterozygote for the A's, homozygous recessive for the B's, homozygous dominant for the C's, heterozygous for the D's. The other parent is big A, big B, little b, big C, little d, two big D's. Okay. And then I say, what is the chance that those two parents can have this kid? So I'm trying to figure out, like, what is the chance that they can have that child there? Heterozygous for all four genes. Now, instead of trying to figure out how to FOIL this and do like a 256 square Punnett square, what you're going to do instead is you're going to do a separate 4-square Punnett square for each of the genes. So, for example, let's say we have a 4-square Punnett square for the A's. So this parent is big A, little a. So the possible gametes are either big A or little a. This parent is big A, little a. So possible gametes are big A or little a. Then you fill in the Punnett square, just as you've learned before. Big A, big A, big A, little a, big A, little a, little a, little a. And then I ask you, what's the chance of having a kid who's big A, little a heterozygote? So that'd be 2 out of 4 or 1/2 chance for the A's. Then I do the same thing for the B's. And I have one parent who is two little b's. The other parent is big B, little b. Fill out the Punnett square. Big B, little b, big B, little b, little b, little b, little b, little b. And I ask, what's the chance I have a kid who's a heterozygote, big B, little b? Well, that'd be 2 out of 4 or 1/2 chance. Then I do the same thing for the C's. Okay, one parent is big C, big C. The other parent is big C, little c. Fill it in, big, 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 little, big, little. And I ask, what's the chance of being a heterozygote, big C, little c? That'd be 2 out of 4 or 1/2. Then I do the same thing for the D's. So for the D's, one parent is big D, little d. The other parent is two big D's. Fill it in, big D, big D, big D, big D, big D, little d, big D, little d. And I ask, what's the chance of being a heterozygote again, big D, little d? So that'd be 1/2. So that's the chance for each of these genes individually. Now, if I want to know what's the chance this kid has all 4 of these for the genes at the same time, I just multiply those fractions. So 1 times 1 times 1 times 1 is just 1. 2 times 2 is 4, times 2 is 8, times 2 is 16. So the chance that these two parents can have this kid is a 1 in 16 chance. Okay, and that's how you'll solve it if you have more than 2 genes. Okay, so now we're going to work on a problem that involves an X-linked gene. So how I want you to do this is I'm going to want you to pause this video and on your own with a pencil and paper try to work through this problem and find the solution. Actually do that step. Try to do it yourself. That's an important part of the learning process for genetics. And then when you think you have the solution, unpause the video and I'll work through it with you. Okay, so when I see a genetics problem that is a word-based problem, which a lot of genetics problems are word-based problems, the first thing I do is I read through the entire problem. And then after I've read through the entire problem, I go back and I reread it and I start drawing out what I'm reading because it helps it to make sense to me. So reading through the problem, red-green color blindness is caused by a sex-linked recessive allele. A colorblind man marries a woman with normal color vision whose father was colorblind. What's the probability that their daughter is colorblind? And what's the probability that their son is colorblind? Okay, so now I've read through the whole thing. I'm going to go back and I'm going to reread it. Red-green color blindness is caused by a sex-linked recessive allele. Okay, both of those are very important parts of information. Then it says a colorblind man. Okay, so I'm going to stop there. And we have a biological male, which means he has an X chromosome and a Y chromosome. And since this is sex-linked, I need to keep track of the sex chromosomes. And it says he's colorblind. Okay, and colorblindness is recessive, okay, on the X chromosome. So that means he would be X, let's say, little b, Y. Now, importantly, this gene is not on the Y, it's only on the X. Okay, so that's a colorblind man. And he's going to marry a biological female, so 2X chromosomes, with normal color vision. Now, if she's normal color vision, she's not colorblind. That means she's either X big B, or X big B, X little b. So right now, I don't know if she's homozygous dominant or if she's heterozygous, a carrier. Okay, so I'm going to write X big B, X dash, because I'm not sure if this dash is a capital B or a lowercase b. Then I continue reading, and it says that this woman's father was colorblind. Okay, so her dad, XY, was colorblind. That was her dad. Okay, now that information now tells us what this dash is. It tells us this dash has to be a lowercase b. Okay, why is that? Because the father has to give something to his daughter. He could not have given his Y chromosome. Since she's a biological female, she doesn't have a Y, which means he had to give his daughter the X. And his X has a lowercase b on it. Okay, so now we know the genotypes of the man and the woman. And it asks, what's the probability that their daughter is colorblind? What's the probability that their son is colorblind? So to solve that, now what we're going to do is a Punnett square. So we draw out a 4-square Punnett square. And we're going to put the possible gametes from the father on the top. So that'd be X little b or Y. Possible gametes from the mother are X big B and X little b. Then you fill in the Punnett square. X big B, X little b, X little b, X little b, X big B, Y, X little b, Y. Okay, and then the question asks, first, what is the probability that their daughter is colorblind? Okay, so if you are wondering about the daughters, then you don't look at the sons. Okay, so we're only going to look at the 2 X's over here. Okay, so these are the daughters. Now, this daughter, X big B, X little b, she is not colorblind. She's a carrier, but she's not colorblind. This daughter, X little b, she is colorblind. So for question A, what's the chance that a daughter is colorblind? Is 1/2 or 50%. Then the next question asks, what's the probability that the son is colorblind? Again, if you're only looking at the sons, do not look at the daughters. So the sons are the XYs. So what's the chance that his son is colorblind? X big B Y, not colorblind. X little b y, he is colorblind. So for question B, what's the chance that his son is colorblind? Is again 1/2 or 50%. Okay, and that's how you would solve that one. Okay, and now let's work through a practice problem that involves codominance, in particular the human blood types. Again, pause the video, try to solve this problem yourself. When you have a solution, unpause the video. A woman who is blood type B has a daughter who is type O and a son who is type A. What is the genotype of the father? Okay, so again, as always, I draw it out to help me understand it. So there's a woman who is blood type B. Okay, and then she has a daughter who is blood type O. And she also has a son who is blood type A. And then we want to know what is the possible genotype of the father. Okay, so based upon what you've learned about the human blood types, if somebody is blood type O, they have to be OO. That's the only possibility they can be. If somebody is blood type B, they can either be BB or BO. Now, since this woman has a daughter that has 2 O's, she had to give something to her daughter, so she can't be BB, otherwise her daughter would have a B. So that means she has to be BO. Now, since the daughter has 2 O's, 1 O comes from mom, that means the other O had to come from dad. Okay, now, they have a son who's blood type A. Okay, well the A could not have come from mom because mom doesn't have an A, which means the A had to come from dad. Now if the A came from dad, this son could either be AA or AO to be blood type A. If the A came from dad, that means mom gave the son the O. She couldn't have given the son the B, otherwise the son would be blood type AB. Okay, so now we know everybody's genotype. The question was actually asking about the father. We figured out everybody. So dad has to be blood type AO. And that's how you solve that one. All right, here's another example problem. Please pause the video. Try to work it out yourself. When you think you have a solution, unpause the video. All right, in cats, black fur color is caused by an X-linked allele. The other allele at this locus causes orange color. The heterozygote is tortoiseshell color. What kinds of offspring would you expect from the cross of a tortoiseshell female with an orange male? Okay, so I've read the problem. Now I'm going to start basically drawing out what I'm reading. And this problem starts to tell you about this particular gene. Okay, and the first thing it tells you is that it's X-linked. Okay, so now we know we have to pay attention to the X and the Y chromosomes. So it tells you that one version of this allele causes the black fur color. So we can have X big B, or X big B, Y, and that would cause the black. Okay, the other allele of this locus causes orange color. So let's say X little b, or X little b, Y causes orange. Okay, now if you notice, I didn't say for black X big B dash. The reason is because the heterozygote is tortoiseshell color. So what that means is if you're X big B, X little b, you're not black or orange, you're tortoiseshell. Okay, so now I kind of made myself a little key, so I keep in mind what I'm looking for. Now it then asks, what kinds of offspring would you expect from a cross of a tortoiseshell female with an orange male? So I have a tortoiseshell female, so X big B, X little b, mated to, okay, a male, XY, that's orange, so that'd be X little by. and I want to know what the kid's going to look like. Okay, so for that, I would use a 4-square Punnett square. So the possible gametes from the father are X little b y. Possible gametes from the mother are X big B, X little b. Then I fill it in X big B, X little b, X little b, X little b, X big B y, X little b y. Okay, now what's inside here are the possible kids they can have. And then based on my key over here, what I find that I have, okay, so 1/4 of the kids are females that are tortoiseshell. 1/4 of the kids are females that are orange. 1/4 of the kids are males that are black, and 1/4 of the kids are males that are orange. Okay, so that's how you solve that one. All right, so once again, pause the video, attempt to answer this question on your own. When you think you have a solution, unpause the video and I'll walk through it with you. Tallness in snapdragons is dominant to dwarfness, while red flower color is dominant to white, and the heterozygous condition results in pink flower color. A dwarf pink snapdragon is crossed with a plant homozygous for tallness and white flowers. What are the genotypes and phenotypes of the F1 individuals? Okay, so let me actually start by making myself a little key. So tallness is dominant to dwarfness. So big T dash is tall. 2 little t's is dwarf in these plants. Okay, then red is dominant to white, but the heterozygous is pink. Okay, so this is an example of incomplete dominance versus complete dominance. So in this case, 2 big R's is red, 2 little r's is white, but the heterozygote looks halfway between the 2 homozygotes, so halfway between red and white is pink. Okay, so that's incomplete dominance. Okay, but now I made myself a little key. All right, so now that I have my key, I'm going to read the question, and then I'm going to draw out what I'm reading. So I start out with a dwarf, okay, which according to my key means they're little t, pink, which according to my key is big R, little r. Snapdragon that's crossed with a plant that's homozygous for tallness. Tallness would be big T dash, but it tells you it's homozygous, so it has to be big T, big T. And white flowers, white is 2 little r's. Okay, and then what are the genotypes and phenotypes of the F1 individuals? Okay, so I want to know what are the kids from this cross. Now this is a dihybrid cross, it's 2 genes. So typically how you would solve that is you would do a 16 square Punnett square. Okay, so let's say we go and we're going to solve this with a 16 square Punnett square. So we draw out our 16 square Punnett square. And then what we have to do is we have to FOIL the 2 parents. So let's say that this one on the left, that's the dad. Okay, we FOIL him first, little t, big R. Outer, little t, little r. Inner, little t, big R. Last, little t, little r. Then we FOIL the mom, big T, little r. Big, that's first, then outer, big T, little r. Then inner, big T, little r. Then last, big T, little r. Okay. Now if you notice, all 4 of these rows have the same gamete for the mom. So technically, we don't have to fill in all 16 square Punnett squares. Everything in this column is going to be exactly the same because all 4 of these are all big T little r. Everything in this column is going to be the same, this column the same, this column the same. So technically, since all of these are the exact same gamete, we don't have to fill in, these 3 columns. We can just fill in that first one, and that will tell us the information. So now let's say we fill that in. Big T, big R, little r. Big T, little r, little r. Big T, big R, little r. Big T, little r, little r. Okay, so in here, what we have is we have 2 of them, this one and this one, that are big T, big R, little r, which is tall and pink. Okay, so that's 2 out of 4, or 1/2 of them are that. Then these 2 are both big T, 2 little r's. Okay, so 2 out of 4, or 1/2, are big T, 2 little r's, or tall and white. Okay, and that's how you would solve that problem. All right, and here's another practice problem. This time it's a pedigree. Try to answer these 3 questions. When you think you have the solution, unpause the video and we'll discuss it together. Okay, so the first question is, based upon this pedigree, is this most likely a dominant or a recessive gene? So in this case, every single time somebody has it, at least one of their parents has it. Somebody has it, at least one of their parents has it. So what that means is it's most likely a dominant gene. Okay, so that's dominant. That's the answer to the first question. Second question, what's the genotype of individual 2,4? So 2,4, so this person right here, it wants to know what their genotype is. Okay, so they're filled in, and since it's a dominant, that means they have to have at least one dominant. So let's say big A dash. And they could be big A, or big A, little a. Now to figure out what the dash is, she doesn't have any kids, so you can't tell by her kids. So you can only tell by her parents. Okay, now the reason she has this big A, the reason she's filled in, is because of her mom. Her mom has the disease, so that means she has a big A, okay, and she passed it on to her daughter. Okay, the dad is an empty square, so that means he does not have the disease, so that means he's homozygous recessive, 2 little a's. Now he had to give his daughter something. He didn't have a big A to give, so he had to give his daughter a little a. Okay, so the genotype of individual 2,4 is big A, little a, or a heterozygote. Third question, what's the chance that the progeny of 2,2 and 2,3 will not have the disease? So in other words, if these 2 have a kid, okay, what's the chance that the kid that they have will not have the disease? That's what we want to know. What's the chance that the kid will not have the disease? Okay, so she's not filled in, individual 2,2. So that means she does not have the disease, so she's 2 little a's. He is filled in, big A dash. His dad had to give him something. All his dad has is 2 little a's, so he's big A, little a. Okay, so to figure out what their kids are going to be, you do a Punnett square. So the dad is big A, little a, this one here. The mom, 2 little a's. Possible kids, big A, little a, big A, little a, little a, little a, little a, little a. What's the chance that the progeny, so their kids, of these 2, so these 2 right here, will not have the disease? So to not have the disease, since it's a dominant, that means they can't have the dominant, so it has to be homozygous recessive. So the chance of that is 2 out of 4, or 1/2, or 50% chance. Okay, and that's how we'd answer those 3 questions. All right, so now let's try a slightly more challenging problem. Again, I want you to pause the video now. Try to work through this on your own with pencil and paper, and then when you think you have a solution, unpause the video. But make sure you attempt it yourself first, because that is an important part of this learning process. Okay, so a male that has a form of dwarfism with normal color vision marries a colorblind woman of normal height. The man's father was 6 feet tall, and both the woman's parents were of average height. This form of dwarfism is autosomal dominant, and red-green colorblindness is X-linked recessive. How many of their sons would be colorblind and of normal height? All right, so as always, what I do is I read the whole problem first, as I just did, and then I go back and I reread it and I start drawing out what I'm reading. Now, one of the reasons I read the entire problem first before I do anything else is because some of the important information might not be at the very beginning. Like, it starts talking about the individuals at the beginning, but if you don't off the top of your head know what type of inheritance this form of dwarfism has and what type of inheritance colorblindness is, then you wouldn't know what that really means. You have to wait until you get to the end where it tells you that this form of dwarfism is autosomal dominant and this form of colorblindness is X-linked recessive. Okay, that's important information that comes at the end. So now that I've read the entire thing, I'm going to go back. So you have a male that has this form of dwarfism. Okay, and it says this form of dwarfism is autosomal dominant. Okay, so that means since he has it, he has to have at least one dominant, so let's say big D. And he could be big D, or big D, little d, so I'm going to put big D dash for now. Then it says he has normal vision, okay, and it tells you that color blindness is X-linked recessive. Okay, so since it's on the X chromosome, now we need to list his sex chromosomes, which he has an X and a Y. He has normal vision, and since colorblindness is recessive, that means he must have the dominant on the X. Let's say big B. So X big B Y. Remember, the gene is not on the Y, it's only on the X. Okay, and then it says that he marries a woman who is colorblind. Okay, so if she's colorblind, okay, remember that's on the X chromosome, and she has 2 X chromosomes since she's a biological female. Since she's colorblind and colorblindness is recessive, that means she must be X little b, X little b. Okay, and then it says that she's of normal height. Okay, this form of dwarfism is dominant, so if she does not have that form of dwarfism, then she must be homozygous recessive, 2 little d's. Okay, then we keep reading, and it says the man's father, so this man's father, his dad, okay, that's what we're talking about, was 6 feet tall. Okay, in other words, he did not have this form of dwarfism. So if he doesn't have the form of dwarfism, that means he cannot have a capital D, so he must be 2 little d's. Okay, and then it says both of the woman's parents, so the woman's mother and the woman's father, okay, were of average height. Okay, so they did not have that form of dwarfism, so they have 2 little d's, both of them. Okay, so what I just did is I just took the information that was given to me, okay, this part here, and I just drew it out. Okay, why? Because drawing it out helps me to understand it. And drawing it out helps me to fill in some of the missing information. For example, I know the man has this form of dwarfism, so it could be big D, or big D, little d. But by writing out this information, I now know what this dash is. This dash has to be a little d. Why? Because his dad has 2 little d's. His dad had to pass something on to him, so now I know he's big D, little d. Okay, so now I know both parents. And then the question is, what is the chance? So we want to know, what is the chance that their son, so X, Y, would be colorblind, so X little b, Y, and of normal height, so 2 little d's. So what's the chance they can have this son here? That's what we're wanting to find out. Now, since this is 2 different genes, this would be a dihybrid cross, and you could foil this and do a 16 square Punnett square. But honestly, the easier way to do it would be to do each gene separately as a 4 square for this particular problem. So let's say I do a 4 square for the D's. Dad had big D, little d. Mom had 2 little d's. So big D, little d, big D, little d, little d, little d, little d, little d. Okay, and I want to know what's the chance that the son is 2 little d's. Well, that would be 2 out of 4, or 1/2 chance for the D's, for that form of dwarfism. Then we do the same thing for the sex chromosomes. Okay, so the dad was X big B Y. Mom was X little b, X little b. So possible kids are X big B, X little b, X big B, X little b, X little b y, X little b y. And I want to know what's the chance that their son, okay, so since we're only talking about the sons, we don't look at the daughters. So what's the chance that their son is colorblind? Okay, well 100% of the sons are colorblind. Okay, so 100%, or 1 out of 1. Okay, now what's the chance that both are happening at the same time? You just multiply these fractions. 1 times 1 is 1, 2 times 1 is 2. So the chance that their son is colorblind is 1/2, or 50% chance. Okay, and that's how you would solve that problem.